Very good afternoon to all of you. Okay, today in chapter 7, we'll be studying some of the important microorganisms. Okay, maybe eukaryotic or prokaryotic, which cause the diseases, plant diseases. Okay, so earlier in this chapter, your bacteria and fungi are also there. My voice is audible?
Yes, sir.
Yes, sir.
Your Adarsh Kumar, Avinash Kumar, Avinash Kumar, Neeraj Kumar.
Yes, sir.
Yeah, my voice is audible.
Yes, sir.
Okay. Okay, Rohit Datta.
Yes, sir.
Yeah, we'll proceed now. Okay.
Okay, sir.
Okay, I told you that today we're studying a chapter that means important uh, your some of the important microorganisms. It may be eukaryotic or prokaryotic, which are responsible to cause your plant diseases. These are microscopic organisms, microorganisms. So in this chapter, your bacteria and funguses are there, but we will discuss because already we have started the bacteria. I have given you the definition and all these things. We skip, okay, all reproduction and your morphology. And fungi will be deal in detail by Dr. C. H. Chaudhary. I think it is started fungi or not. Rohit Datta,
Yes, sir.
Jyoti,
Yes, sir. He has started.
fungi started or not?
Yes, sir.
Yes, tell loudly.
Yes, sir.
Tell loudly. Fungi, you, Saraj, started or not?
Yes, sir.
Started.
Yes, sir.
Okay. So, uh, we'll be here in this chapter, I will not discussing the bacteria as well as fungi because already bacteria I have dealt, okay, wherein I have given you the general morphology, reproduction, and all such things, and uh, fungi will be taken by the Dr. C. H. Chaudhary. So, other than that, what are the microorganisms are here? Of course, one more was there, that is phenogrammic, your higher plants. That also I will not touch because it will be also taught you by Dr. C. H. Chaudhary only. Okay, so it will be simply a repetition. So except you might have got the syllabus. Is not it?
Yes, sir.
You got the syllabus?
Yes, sir. We have the syllabus.
Course breakup, have you got?
Yes, sir.
Yeah, tell loudly. So today I am starting the chapter 7. You can see here we will not study about the bacteria because already covered bacteria, fungi and phenogrammic higher plants. It will be taken by Dr. Chaudhary. So other than those, which microorganisms will be left that we will study today. So among those, starting with the Fastidius vascular bacteria. Okay. So these are also commonly known as RLO. RLO means, anyone can say, Jyoti, what do you mean by RLO? Rohit Datta.
Yes, sir.
Jyoti Jha.
Sir.
What do you mean by
Risk
RLO?
like organisms.
Yeah, yes.
Risk-like organisms.
What? Tell loudly.
Sir, risk-like organisms.
Rickettsia. Yeah, Rickettsia like organisms. Okay. So this fastidious vascular bacteria, it causes your plant diseases and it cannot be grown on the simple culture media in absence of host cell. That means these are obligate. Generally, these are obligate. Okay. So in the laboratories, generally for growing the bacteria, you are using the nutrient agar or you are using the Kingsby media or in case of broth, These are the solid media. In broth, you are using the succinic media. These are the general media where the bacteria are grown. But in this, in this type of your artificial media, which are used in laboratory culture, okay, these microorganisms cannot be grown. Okay, it needs the host cell, living host cell. So these are generally the rod cell. That is, my voice is audible, Rohit Datta.
Yes, sir.
Okay. If sometimes it breaks, then you can call me on my mobile. Okay. And
Okay,
Jyoti,
sir.
Yes, sir.
okay. Your voices are very, as if you have not taken food. You have not taken lunch.
Sir, I have taken.
You are in the fasting, you people. Navaratra. That is why your voice is not coming. Anyway, it is a rock set. Size is 0.2 to 0.5 micrometer in diameter and 1 to 4 micrometer in length. Okay. It is bounded by cell membrane as well as cell wall. Okay. It is bounded by both cell membrane as well as cell wall. It doesn't have any flagella for its movement. Okay. It doesn't have flagella. Cells are usually undulated or ribbed. Okay. Cells in the you'll find that is the undulated or ribbed. So, like your limited vascular so in the culture media, when it is grown, you can see the cells that the culture or growth as undulated or ribbed. And almost all the fastidious vascular bacteria are gram-negative in nature. Okay. Of course, there are some few exceptions you will come to know in later part of our discussion. These are sensitive to several antibiotics such as tetracycline and penicillin. Control measure, how to control these fastidious vascular bacteria. So hot water treatment is found to be the most effective, okay, in controlling such bacteria, that is FPV, fastidious vascular bacteria. So here entire plant or propagative organ, suppose some cuttings if you are taking, budding, cutting, okay, or grafted part. So this should be treated in hot water treatment at the temperature that is 45 Celsius. More than this, that is 50 to 58 degrees Celsius. Okay, so at this temperature, the nature, so low temperature, it is treated at low temperature, and just I will tell you. I will tell you. Okay, okay. No. We have sent more. I will just tell him to call. Okay, madam. So, no. He has sent. I think he has sent more than kg, near kg. I think. I will tell. Yeah, mixer का. Hmm. Oh, check. Yeah. Oh, I will tell him to call. Okay, madam, to you. Okay, madam. Okay. Yes, madam. Please. Hello, Khalid. Khalid, you just called Radhe, madam. I'm in class. She was telling that fennel seeds are only 100 gram. I told her it is more. We have sent. So once you confirm, okay, you talk to Radhe, madam, Calicut. You are having the number. She is not having your number. I think you are having. Oh, so I will just now I'll WhatsApp her number. Okay. I'll WhatsApp just to cross check what is happening. She is telling me 100 gram seed they have received. I don't know it is near kg, 1 kg near. Hmm. Just you check it. Okay. I am sending you the Radhe madam number WhatsApp. Okay. Okay. Uh, Rohit Datta. Rohit Datta. Rohit
Yes, sir.
Datta. Just 1 minute. Sorry. Some important call came from Calicut, Kerala regarding seats. Just a minute. I will continue. Just a minute.
Okay, sir.
Okay. Khalid, just now I sent. Did you got? If, okay. You just check it. Okay. 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 Sorry, students. So I was telling some succulent plant, and it is treated at your lower temperature, that is at 45 to 50 degrees Celsius, for lesser period of time, that is 2 to 3 hours. For some hardy cuttings like citrus, okay, hardy cuttings, uh, okay. So it is treated at 50 to 58 degrees centigrade for 4 to 8 hours. So example, it is very much effective for grape vine Pierce disease, because this disease was first reported by Pierce. That is why it is known as grape vine Pierce disease in grape. And another disease is sugar cane ratoon disease. Okay, sugar cane ratoon disease. So this fastidious vascular bacteria, this is of two kinds. One is your phloem limited fastidious vascular bacteria, another one is Jalen limited fastidious vascular bacteria. So the your phloem limited vascular bacteria, that means these bacteria are present or they are limited in the phloem vessel of the plant. Where in case of Jalen limited bacteria, these are inhibited or they are found or they are present or they in fact the Jalen vessel of the host system. Okay. So this Jalam Limited or Fluem Limited, it is completely based on their presence, whether it is in Jalam or Fluem, or site of infection is Jalam or Fluem. Okay. So coming first to the Fluem Limited, first it is vascular bacteria. This was first observed in 1972 in Fluem of clover. Okay. And periwinkle plant. Periwinkle, you know, this is a flowering plant. It is very commonly available everywhere. Do you know periwinkle? Rohit Datta?
Yes, sir.
How it looks like?
Sir, it has flower of white and pink colors.
Anyone can explain the periwinkle plant?
सर पिंक कलर छोटा छोटा हरा होता है एक बार
Voice is not clear. What is commonly it is known as? In common name?
सर सदाबहार
Sadabahar or either white, mainly the pink color. There are 4 petals in the flower, pink color. Okay, and now white color is also available, and it also grows sometimes wild also. Okay, periwinkle. So it is so it was first observed in 1972 in the phloem of clover as well as the periwinkle plant affected with the clover club leaf disease. The disease was known as clover club leaf disease, and later it was also found in citrus plant affected with greening disease. Okay, citrus greening. These are mostly gram-negative, rigid rods, non-motile, bounded by double membrane. Like peptidoglycan is absent. Like it is present in most of the bacterial cells, but here peptidoglycan is absent. Disease symptom, it is leaf stunting and clubbing. This symptom is leaf stunting and clubbing. That means leaf becomes, your size is reduced, and the tendency of leaf or to grow towards the centrally part. Okay, it will not spread. It will make a broom-like structure. So shoot proliferation and itches brooms are also the common symptom. Okay, shoot proliferation, that means from a node there will be excessive your emergence of shoot than the normal from the node. Okay. Then another symptom is greening of floral parts. The floral parts will remain green in color. Factor is your leaf hopper, psyllid. Okay, these are the insect vector, leaf hopper, psyllid. These are also very slow moving insects, and it will find that it will be get stuck in the stem or trunk of the citrus plant, also in the mango plant. These are the very slow moving insect, psyllid. Doders, it is your anagrammic higher plants, parasitic plants, and of course mechanically it is also transmitted by grafting. During the grafting, mechanically it is transmitted. Example is cytoplasmic disease. Then clover club bacteria. Okay, this bacteria can pass from other leaf paper to its eggs. Such type of transmission of this your bacteria. Okay, like limited bacteria that from one generation of sac to another generation of sac through eggs. It is known as transferarial transmission. Transferarial transmission of your this means your causing agent or microorganism. Okay. Bacteria. Okay. Next slide, can you move? Limited bacteria. Coming to the bacteria, one year later, in 1973, it was observed in Jalam cells. Cells indicated as Jalam limited. It is observed in Jalam of the plants. It affected with your disease and alpha to the disease. Alpha is the plant, which is the disease. These are also negative. Action is there. Action Vascular can rattle and tendency, which is positive in nature. Okay. And also with this, if you're not translated, this is your it is translated mechanically only. Like wrapping on all these things. Okay. So the process non-support forming, non-life, well-defined layer cell wall and plasma membrane. So cell membrane is the same. So in middle of that plasma membrane is present. Cell wall and plasma membrane. So unlike millimeter area, I told in the starting, all millimeter vascular area. And now compact like media. Okay. Compact media, that media, artificial media, system, ethereal, growth regulator. Okay. Or that means every system runs on growth regulator. So the artificial media like media, other media, it is very temporary. Some your referral growth regulator. Those media can be and these are the only sets of translated. Okay. Apply your translated by your daughter. Not it. Daughter is your it is translated by developing the set of papa. It's the bug. Okay. So that can acquire and transfer bacteria in less than 1 hour. So this is period is also managed in less than 1. This is the period of the time required to acquire and transmit the bacteria. Okay. This is also managed. This period is also managed. Anyone? Or anyone?
Infested period.
Yes.
Infested period.
No. It is acquisition period. It is acquisition period. You are from Sasa.
Madhubani, sir.
Madhubani. What is your name?
Amar Prem, sir.
Amar
Prem.
If you come from Muzaffarpur, Amar is the line of Muzaffarpur. I say, Amar is
Yes, sir.
Yeah. But anyway, so say your acquisition certificate. Okay. That means that period required by a paragraph sector to acquire the bacteria. This is use this case of virus. Okay. But we talk here about only bacteria. This is the idea by sector to acquire the infected organism or the bacteria and to transmit next to the your okay. So less than 2 hours. That means evaporated to 2 hours. Just after 2 hours, it can be transmitted to the other problem. Okay. So symptom, the marginally cases. That means your rate of the tissue from the marginal lips, necrosis is there, strengthening of one surviving, and after that generally there will be decline. Okay. Decline of the whole plant. There will be decline of the common cellular behavior, decline from the bottom, slowly. That's what cycle irrigation process is. Okay. And the plant will appear yellow color, but the irrigation is there will be patches on the leaf yellow and green patches. That is why we know fluorosis. Then only T okay, and the T is a blue after the only hormone in this scorching the hormone. Lung is scared, so it is a severe form of scorching. Okay, burning is a severe form of scorching. These are all called bacteria. Okay, compound bacteria. Gallella fastidiosa. Gallella fastidiosa. Of course, there is an exception. Okay, sugar cane is told starting of slide. So this is transmitted by a disease called by clavicle services and it is gram bacteria. And also this is known to be transmitted mechanically only. Sugar cane is the exceptions. First is that it is gram positive. Gram positive, unlike the other denominated bacteria or denominated bacteria. And secondly, it is transmitted mechanically. Okay, so gram burning. Okay, cutting also. So should I write? Should we receive the next slide?
Yes, sir.
Okay. Slide was there are also synonym disease molecules. Okay. I think my brain is a subject and free molecules. So these molecules, the mycoplasma. So mycoplasma is mycoplasma that is causing disease in plants. Mycoplasma. So there are molecules that affect plant and can or mycoplasma, and it is observed in disease plants and insect vector. It is also observed in disease plants as this is molecules mycoplasma are also observed in the insect vector. Characteristics: it lacks the cell wall. Okay. Cell is bounded by the membrane. It is cytoplasm, ribosomes, and strands of nuclear material. Strands of nuclear material. It is difficult to avoid or irregular blood flow. That means it is several or blood vessels also. Generally, it is a sector of it is mycoplasma is found in the left tissue. Majority of them are transmitted by leaf purpose, some by cylids, and also plant purpose. Okay. It also grows in the electric canal, namely the salivary gland, and intracellularly in the body organ of their sex factor. That means they also inside the body, in the electric canal only, the salivary gland or the body. In that purpose, yellow papaya, you find that the yellow of the papaya and then these are coming shorted and when you take it among the leaves and you press it, breaking sound will come. Then the yellow papaya and papaya die back, passes through which is yellow, yellow, yet decline, crack yellow, little yellow coconut. The little coconut, that means the yellow in the appear in the plant and with the plant die. Okay. So this is regarding mycoplasma. So we move to the next. Mycoplasma.
Yes, sir.
Okay. So this is mycoplasma. Okay. It is also a kind of mycoplasma. So there are biomorphic mitochondria. When it is exposed to unfavorable environment, it changes its shape. Okay, it becomes and behave like chitin. We have biomorphic wireless microorganisms that are present in the web of disease plants like your cytoplasm. Sorry, they are helical in nature and what different kind of microplasm. In these are your microplasm is spherical, wide, tubular, but they are helical in culture. And there is also thought to be a kind of microplasm only. Same size, spherical, slightly wide, 100 to 240 nanometer, are larger in diameter, helical and non-helical filament. Okay, it may be helical or it will be non-helical filament. 1.20 nanometer in diameter and 2 micrometer long. Mostly helical forms in liquid media. Helical form of the in case of when it is grown in liquid media. Like one. It can be on the set matter and it can be cultured on nutrient media. Okay. So like plasma, it can be cultured on nutrient media. Of course, this media should be an enriched one. Okay. So this media is also known as enriched media, auxiliary media. This media is enriched by hormones. Okay. Those regular minerals, these are known as your oxygenic media. Okay. These are the media used for growing of some of the bigger parasites. It multiplies by fusion. It lacks flagella. It lacks two-cell wall and surrounded by a unique membrane. Helical filaments are modified, moving by a slow convolution of filament and probably by a rapid rotary or slow motion in the helix. So its movement is like probably by a rapid rotary or slow motion of the helix. If someone of well was an specific channel, there was rattlesnake is seen or groups rattlesnake is anyone can say rattlesnake is seen in the television in the groups. No one has seen the rattlesnake in television. Rattlesnake.
Yes, sir.
हमारा फ्रेंड
रेजल्ट्स में देखा है सबके रिजल्ट्स में पाया जाता है कि मतलब मुझ पे कोई लगा हुआ जैसे साबित करते हैं
वॉइस इज नॉट क्लियर
सर रेटिना का टेल का मूवमेंट बहुत फास्ट होता है सर।
देखा है आपने
ये सर वो देखा है बहुत फास्ट मूव करता है और उसका टेल
सबसे हेलिकल स्ट्रक्चर ओके इस बात sometimes there is a video thing okay this channel they are sometimes so this rattlesnake is very fast being move helical structure okay so this only on particle area it appears electrical right like or granular. So when it was in the media, it appears like as if some appearance like right eggs or granular. Okay. It is some media. The growth is not it is resistant to penicillin and inhibited by tetracycline. Example, citrus, apple disease, brittle root disease, ulceratis, congestion disease, then disease in periwinkle. Disease in periwinkle. Okay. This periwinkle is present in every house, every village. Many see periwinkle plant. Small plant, flowering plant. Okay.
So what type of disease, sir?
Disease.
Disease.
Yeah. See, plasma. Basically, we will call this disease yellowing symptom. Okay. Followed by your stunting of the plant. Plant will get stunted and yellowing. Okay.
Okay, sir.
So should we how much time?
Yes, sir.
The next slide.
Yes, sir.
So virus. Okay. Viruses will be out there. It will also there in there by our especially I give you some idea why viruses. So viruses is living or not living? So the viruses, no one can say whether it is living or not living.
Structure.
It is living and
Inert structure.
crystalline. So several it becomes crystalline in nature. Okay. And this tobacco virus, you know, if it gets the favorable temperature and the crystalline form, it can remain viable or inactive after 50 years also. Okay. But for 50 years, it will be a dormant state, in the viable state, when the favorable condition will come, it can cause the disease. Virus. So up to 3 years, it can remain inactive. Okay. Half century. So these viruses are submicroscopic. So these viruses can be observed by microscope or not.
Yes, the microscopic can be observed by electron microscope.
Then
The microscopic.
Yes, sir. Microscopic.
microscopy.
Submicroscopic, sir.
What kind of microscope is visible? Visible or or is it microscope or ultraviolet microscope or fluorescent microscope? Which type of microscope can be observed? Well,
Yes,
these viruses can be observed by what kind of microscope? So many I told you, stereo microscope.
microscope.
Yes, electron. Good. What type of electron microscope? Have you heard the name of SEM? SEM, SEM.
Yes, sir.
No,
What do you mean by SEM?
Scanning electron microscope.
Yeah, Aniket. So,
Yes, sir.
so long you are not responding. You are sleeping from starting
No,
of class.
Sir, my voice was not unable to reach you.
What happened to your voice? You have not taken food.
No, sir. There is a problem with my mic.
Oh, Mike. Now it's okay.
Yes, sir.
Yeah. So Same, that is scanning electron microscope. Okay. Coming to the viruses, these are some microscopic obligate parasites. Obligate means, you know, what do you mean by obligate?
True, sir.
Obligate means?
It's a true parasite.
Means what
Not
this
change the
means.
behavior.
So it can be grown in the culture media or not?
No, sir.
No, sir.
No. Okay. Obligate. They need living host. Is not it?
Yes,
Consisting
sir.
of nucleic acid. So this nucleic acid may be RNA or DNA, and protein code that replicate inside the living cell using the host cellular synthesis mechanism. You might have bacteriophages. You might have heard the name of bacteriophages. Okay. So these viruses, they are killing the bacteria, and they use the mechanism of bacterial cells for its replication. Earlier, it was also called as poison or venom in Roman times. Earlier, it was also called as poison or venom in Roman times. So, first viral plant disease, it was known tobacco mosaic virus, and it was first, okay, observed by Adolf and Mayer, or identified by Adolf and Mayer. Then later on, the Bejerin called this virus, it as contagium vivum fluidum. That meaning is that contagious living fluid. This living fluid, if it comes in contact, so it will cause infection. That is contagious living fluid. Okay. So nature, so these viruses are acellular, they are submicroscopic and intracellular. They lack the lipid membrane system and energy production. It lacks the mechanism of energy production also. They use host machinery for their replication. As in case of bacteriophages, these virus particles use the replication machinery of bacterial cells after killing them. Then viruses will deal in detail in the virus chapter. So this viroid. So these viroid are smaller or viruses are smaller?
Steroids.
And virion, so of course these viroids are smaller than virus. So until 1971, it was unknown. So for the first time, Diner, okay, he observed this viroid, or he identified this viroid in potato spindle tuber disease. Okay, so when this viroid affects the potato plant, the potato tuber becomes spindle in shape. Okay, tuber shape, spindle in shape. So it is caused by these viroids are the non-encapsulated RNA particle. Okay, these are small, circular, single-stranded, low molecular weight RNA molecule with extensive base pairing that can infect plant cells, replicate themselves to cause disease. So here the base pairing is more than your virus particle. So transmission of viroid, it takes, in case of virus also, mainly the transmission is taken by your insect vector, and of course by your plants, okay, mechanically, then fungi also, okay, by human. So these are the mode of transmission of virus particle. So here also the viroid, they are transmitted mechanically, sap carried on the hands, tools, vegetatively propagated method. In case of vegetatively propagated method also, these viroids are transmitted through the cell sac. Example, coconut kadang disease. Okay, so this disease, the symptom of this disease is a slow decline of the plant. And the plant will die, and at last, our leaves will also fall down after drying, and the plant will stand alone as if telephone post. Okay, only single trunk would be there in the field with no leaves or hanging leaves, giving a resemblance of your telephone post. In case of coconut karang disease. So coconut karang disease is caused by coconut karang viroid. Okay. The next disease example, citrus exocotis, then chrysanthemum stunt. So chrysanthemum is a flowering plant. So if it attacks the flowering plant like chrysanthemum, the plant becomes stunted in growth. It unable to grow normally. So we can see some of the differences in virus and viroid with respect to nucleic acid. In case of viroid, it lacks protein port and it exists as free RNA. In case of viruses, it is enclosed in a protein port. Okay, it is not naked. Coming to the size of RNA, in case of viroid, it is 250 to 370 base pair. In case of viruses, it is 4 to 20 kilo base. Molecular weight of RNA, in case of viroid, it is 75,000 to 1,20,000 Dalton, whereas in case of viruses, it is 10 lakhs to 1 crore. So these are some differences between the viroid and viruses with respect to their nucleic acid, size of RNA, and molecular weight of RNA. Could you move to the next slide?
Yes, sir.
Yeah. Next means this will be the last slide only. So, algae. Algae also causes disease. Okay. Sometimes in examination, it may be asked that is red rust of tea is caused by an ... I repeat, red rust of tea is caused by an ... What will be the answer? Caused by an
Sir, algae.
algae. When N is there, algae. Mind it. Don't write fungi or bacterial algae. So these are eukaryotic halophytes, not prokaryotic like your bacteria and all this. So these are eukaryotic halophytes having chlorophyll. Algal cells may be spherical, rod-shaped, barrel-shaped, club-shaped, spindle-shaped, etc. Okay. The shape is different, right from spherical to the club-shaped and barrel-shaped. It contains a well-defined nucleus, chloroplast, mitochondria, vacuole. Okay, the vacuoles this keep preserve the food material for the your organism. Oil droplets, starch grain, etc. Some algae move by flagella and some do not have any locomotion. That means wherever it will come, okay, it will be there. They don't have any mechanism of its own locomotion. Okay, but some algae are there which can move through flagella. Example: red rust of tea caused by Cephaloleurus parasiticus, and this red rust of coffee, it is caused by Cephaloleurus coffee. So, should I proceed to the next topic? Last and next topic. Any doubt from this person? Otherwise, proceed to the next heading, that is protozoa. Protozoa. Protozoas are mainly causing disease in humans, that is your stomach related or alimentary canal related diseases. Okay, these are the waterborne organisms. Okay, waterborne diseases they create. In case of plants, okay, it is first reported by Lefont in 1909 in Mauritius, in In Liliaceous plant, Euphorbia. Euphorbia is the family, it comes under Liliaceous plant. Mostly these protozoas are one-celled macroscopic organisms, generally motile, and have typical nuclei. It may live alone or in colonies and may be free-living parasite. It may be free-living or it may be parasite. They move by flagella or by pseudopodia or by the movement of cell itself. Okay. Any doubt? Protozoa, Rohit?
Yes, sir.
Protozoa, so they may be free living or they may be parasite also. Okay. So they move by flagella, by pseudopodia, or by the movement of cell itself. Example, common is your phloem necrosis of coffee, and it is caused by Phytomonas leptoviscera. In coffee, phloem necrosis disease is caused by Phytomonas leptoviscera. So where the death or killing of phloem vessel appears, or necrosis, death of the phloem vessel appears, and ultimately the plant wilts and get killed. So this is all about the microorganisms, important microorganisms which causes the plant disease, except bacteria, fungi, and phenoloramic fire plants. Okay. Any doubt?
No, sir.
Rohit Dutta,
Yes, sir.
any doubt? Jyoti?
No.
No, sir.
Yeah, tell. Amar Prem,
Yes, sir.
any doubt?
Sir, there is Hemilia vestatix, which also cause the
Red rust
of
of tea. Red rust of tea, algal disease.
Okay,
Is not it?
Hemilia vestatix, sir.
Yeah. Is not it?
Yes, sir.
Yeah. So you tell.
Sir, but in the PDF which is given by C.S. Chaudhary, sir, here written, sir, Hemilia vestatix, sir.
Red rust of tea.
Sir, but C.S. Chaudhary PDF mentioned red rust of coffee is done by Hemilia vestatix.
Red rust. Hemilia vestatis. You see, Hemilia vestatis is also sometimes your role of these organisms is not clear. Sometimes with the red rust, both your infection, that is your combined infection is found there. So it is not very clear. Both the organisms are also found in case of your this red rust. Sometimes your Hemilia also and your cephalolurus also. But cephalolurus mainly it causes in case of tea, predominantly it causes red rust in tea, and red rust in coffee is predominantly it is caused by Hemilia vestatix, but of course to some extent also your cephalolurus coffee also is responsible. Okay.
Okay, sir.
Predominantly it is caused by Hemilia vestatix. Okay.
Okay, sir.
Then anyone? All students, you give your attendance in the chat box.
Okay, sir.
Give attendance in the chat box.